I'm very delighted to be in front of you once again. In the previous session, we discussed about what happens to the food particle in the initial part of small intestine, that is duodenum. After the partial digestion of food particle in the duodenum, the food is pushed into the remaining part of small intestine where intestinal glands are present. Intestinal glands secrete intestinal juice. I repeat, intestinal glands secrete intestinal juice. The enzymes present in intestinal juice are disaccharidase and peptidase. Enzymes are disaccharidase and peptidase. The function of disaccharidase is it converts complex carbohydrates into simple carbohydrates. Disaccharidase converts complex carbohydrates into simple carbohydrates. Let us see some examples of disaccharidase. Maltase. It converts maltose into glucose. I repeat, the name of the enzyme it is maltase. It converts complex carbohydrate maltose into glucose. Then second one is lactase. It converts lactose into galactose. The name of the enzyme is lactase. It converts complex carbohydrate lactose into galactose. Third one is sucrase. It converts sucrose into fructose. The name of the enzyme is sucrase. It converts complex carbohydrate sucrose into fructose. I hope you understood what are the different types of disaccharidase. The function of peptidase is it converts peptides into amino acids. I repeat, peptidase converts peptides into amino acids. By the action of the enzymes in intestinal juice, the digestion of food in the small intestine is completed. The byproducts formed during the digestion are glucose, amino acid, fatty acid and glycerol. I repeat, the byproducts formed after digestion are glucose, amino acid, fatty acid and glycerol. There are other components which does not undergo digestion. They are water, roughage, vitamins and minerals. The next topic what we have to discuss is about absorption. Along with the process of digestion, small intestine helps in the process of absorption too. Let us see how far the structure of small intestine is suitable for the process of absorption. are some specialized structures in the small intestine called villi. Villi, it is singular, villus, it is plural. Villi are small finger-like projections seen on the wall of small intestine. I repeat, villi are small finger-like projections seen on the wall of small intestine. They increases the surface area for absorption because millions and millions of villus are present on the wall of small intestine. More number of villus present on the wall of small intestine increases more area for absorption. Now let us have a closer look about the structure of villus. Villus are covered with a single layer of cells. I repeat, villus are covered with a single layer of epithelial cells which help in the process of absorption. They also contain blood capillaries, lacteal and lymph capillaries. They also contain blood capillaries, lacteal and lymph capillaries. The branch of artery, when it enters into the villus, it divides into capillaries. And we all know that capillaries reunite to form the vein. So this is the branch of artery and this is the branch of vein. Similarly, the lymph duct, when it enters into the villus, it divides into numerous lymph capillaries. The lymph vessel found in the villus, it is known as lacteal. The lymph vessel found in the villus, it is known as lacteal. Now let us see what is the function of capillaries and lacteal. Capillaries absorb glucose and amino acids. I repeat, the blood capillaries absorbs glucose and amino acids. Lacteal absorbs fatty acid and glycerol. Lacteal absorbs fatty acid and glycerol. Those nutrients which are soluble in water are absorbed by capillaries. Glucose and amino acid, it is soluble in water. So it is absorbed by blood capillary. The byproduct fatty acid and glycerol is insoluble in water. So it is absorbed by the lacteal. So insoluble substances are absorbed by lacteal. About 90% of absorption of nutrients and water takes place in the small intestine itself. Hope everyone understood today's session. Thank you.